Hey everybody, I hope you're doing well and had a good spring break. I wanted to hop on here and do a quick lesson on DNA and DNA replication because if we can get through these next 3 chapters, then we should have everything covered that's fair game on the exam, which is on May 18th at 2 o'clock, and we'll talk more about that later. So let's talk a little bit about things we may have learned in regular biology. So I taught you guys about Watson and Crick and Franklin and Wilkins. Linus Pauling, you guys probably haven't heard of. He actually was the first one to kind of figure out that DNA should be a spiral structure, but they weren't sure if it had 2 strands or 3 or 1 or however many. So remember, Franklin was the lady that took x-rays, and Wilkins took one of her x-rays because they were arguing and gave it to Watson and Crick. And then when Watson and Crick saw her x-ray, they immediately figured out the structure of DNA. So these 3 ended up getting the Nobel Prize because remember, Franklin had died from ovarian cancer before that. Okay, so if you haven't already, I do want you to check out the 2 videos that I posted for you. One of them's on Griffith's Experiments. It's a cartoon of what Griffith did to figure out that genetic material can transform. And then Hershey-Chase Experiments, these pair also figured out that genetic material had to be what was being passed on and led to the structure of DNA that they figured out in the 1950s. Okay, so speaking of the basic structure, remember all nucleotides, whether they are DNA or RNA, have 3 parts to them. They have a sugar. For DNA, it is deoxyribose. They have a phosphate group. And then they have a nitrogen base. And remember, those 4 nitrogen bases are thymine, adenine, cytosine, and guanine. Again, you don't have to know the names, you just need to know the letters and what pairs with what. And we'll talk about that in a minute. I do want to show you something new in here. There is a 3' end and a 5' end, which that will not become important until DNA replicates itself, and I'll show you that later on in this video. But I just want you to notice why it's called that. Here is the 3rd carbon in the sugar, and the hydroxyl group is attached there. And then the phosphate group is attached to the 5th carbon. So that's why it has a 3', 5' end. All DNAs are this way. Okay? All right, so remember that they are Chargaff, I'll get it right in a minute, figured out that the rule is A always pairs with T and C always pairs with G, and he figured that out with percentages. So if A was 40%, T was automatically 40%, and then the C and G would split the remaining 20% at 10% each. So that's how he figured all that out. Okay, now another new term that you guys need to know, A and G are specific nitrogen bases called purines, and if you'll notice, they are larger nitrogen bases because they are 2 rings together. Okay, the other ones, the C and the T, and then of course U of RNA, are called pyrimidines, and they are just 1 ring of carbons. They don't have that extra ring there. And you have to know that for some particular reason. So, and I have to look back because I can never memorize that. Anyway, remember A pairs with T, C pairs with G. A purine is always going to pair with the pyrimidine and vice versa. Okay, and they are held together by hydrogen bonds. And remember, hydrogen bonds are pretty weak bonds, so we can replicate DNA easily. Okay. All right, so let's talk about replication a little bit. Remember, the original strand is going to serve as a template for the new strand. That way it cuts down on errors. Okay, it's called semi-conservative. So that basically means the 2 new DNA strands that are created, one part is brand new and the other part is old. And that helps cut down on mistakes. So the only original DNA strand you had is the one that was created when sperm and egg united to create you. All of your other ones are semi-conservative. Okay, there are tremendous number of enzymes that do work in DNA replication. So just a couple things I want to point out to you. It's very fast. We can't even show you how fast it is in terms of a model or anything because they can add 500 nucleotides a second. And it would only take a few hours to copy all of our 6 billion base pairs. And usually it's pretty accurate with only making one mistake in a billion. So that's pretty good. Okay, all right, so I want to show you a picture of this. So with something called a replication fork and things like that. Okay, let me see where I want to start here. All right, so you know what a fork in the road is, right? It's where like two different things go in a different direction. Okay, because DNA is antiparallel, we can only add nucleotides to the 3' end of DNA. Remember where that hydroxyl group is. We can't add it to the phosphate group end. Okay, so they have to be added here or here, nowhere else. So that makes it difficult for the two separate strands. So we have one strand called the leading strand and a second strand called the lagging strand, and the way the nucleotides are added is very different. So the leading strand is easy because all you're doing is adding C's and T's and A's and whatever in a row constantly. The lagging strand has it a little bit more difficult because we can't add it at the replication fork. We have to add it at the other end. So two enzymes that you need to know are DNA helicase and DNA ligase. So remember how DNA is a double helix? Well, what this enzyme does is basically unravel the DNA strand. So it takes it from being a twisted double-stranded molecule to looking like a ladder. And then ligase is an enzyme that's going to attach the nucleotides together. So think of it like super gluing them together as they're added. Okay? Now, a Japanese guy named Okazaki came up with the term for this. Okazaki fragments are little DNA pieces that are added to the lagging strand. Remember we said we can't do it piece by piece by piece by piece. We have to do it in little chunks. So refer to the Bozeman video for that. It's a much better description and picture of it. Okay? All right, so here's your original DNA strand. Okay, so notice this is the leading strand, and then the leading strand and the lagging strand. So we can only add them on the 3' end. So here we're going to add them A, T, T, G, C, whatever. Here we have to add them in short 3 to 5 DNA base pieces in little chunks. Okay, so the leading strand actually goes much faster than the lagging strand. And I'll show you all this piece by piece. Okay, so here are some of the enzymes you might have to know, and little binding proteins and different things. I'm going to get you guys to write those down in a little while. Okay, so here's a better picture of that. So this topoisomerase is going to hold basically the DNA strand while it's being untwisted, kind of like it's holding it down. Okay, you've got these little proteins here that are going to kind of stabilize the unwound strands, and here's that enzyme helicase that is actually unwinding the DNA strand as we go. Okay, so helicase unwinds. I'm going to run through this step by step. All right, molecules of binding proteins are going to stabilize the two strands that are unwound, kind of like holding them down. Okay, the leading strand, DNA polymerase, is just going to continuously add A's, T's, C's, G's in the correct order. All right, then the 5th Okazaki fragment is added on the lagging strand. Okay, then we're going to add the 4th Okazaki fragment. So see, it goes backwards. 5, 4, 3, 2, 1. Then it's going to unwind it some more. 5, 4, 3, 2, 1. All right, puts the nucleotides in, kind of seals them together. Okay, so things are much more difficult for the lagging strand. So what I'd like for you to do at this point is please, please, please watch Bozeman's video on DNA replication, because he can do a much better job than I can, especially from here at home. He does it pretty well. And then if you can, try to complete some of the things that I've attached to see if that makes a little bit more sense for you. And then also, please keep working on the Unit 6 stuff that I posted on AP Classroom after we finish some of these other lessons. So you'll be ready. And I will definitely get another lesson ready for you for this week on transcription and translation and things like that. Hope you guys are well. Please email me if you have any questions.